Hello, and welcome to Virtual Investor Conferences. My name is Cecilia, and on behalf of OTC Markets, we're very pleased you've joined us for our next live presentation from Sigyn Therapeutics. Please note you can submit questions for the presenter in the box to the left of the slides. You can also view a company's availability for one-on-one meetings through the Scheduled Meetings tab found on the conference platform. At this point, I'm very pleased to welcome Jim Joyce, co-founder and CEO of Sigyn Therapeutics, which trades on the OTCQB venture market under the symbol SIGY. Welcome, Jim.
Cecilia, thank you for the introduction. Before we get started, I want to bring your attention to our forward-looking statements. These statements are also available under the presentations button on our website, sigyntherapeutics.com. With that said, we invent and develop blood purification therapies. Our therapeutic candidates include the immune prep platform to enhance the performance of immunotherapeutic antibodies. These are the top-selling drugs in cancer, which is the number 2 cause of death in the United States. Chemoprep is optimized to improve the targeted delivery of chemotherapy. And because the delivery of chemotherapy is insuperior, ChemoPure was developed to extract off-target chemotherapy from circulation to mitigate treatment toxicity and to reduce health consequences that are long-term issues. And our lead product, Sigyn therapy, addresses infectious disease orders that are not treatable with drugs. And I think you'll learn from our presentation that we have an early clinical and commercialization opportunity within the dialysis industry. All of our products are supported by an accelerated development strategy that reduces our clinical risk and offers to save millions of dollars in development costs. The immune prep platform leverages the use of pharma-developed antibodies that have already been clinically tested and market approved. Our strategy to reduce the toxicity of chemotherapy and to improve its delivery, it's based on repurposing a previously developed technology that has established manufacturing in place. And Sigyn therapy, it incorporates, this is very important, it incorporates components that are already scaled. The scaled manufacturing is in place to address infectious disease disorders. And this is an area where we have a lot of relevant experience. Previously, I was the founder of Athlon Medical. This is a company we grew from single shareholder startup to NASDAQ company with over 8,000 shareholders and a peak market value of $200 million. I should point out, as this is an over-the-counter virtual market, we started off as an illiquid stock on the OTCQB, and then we eventually transitioned to having a security that on some days would trade millions of dollars of shares. More importantly, while I was at Athlon, we developed the Athlon Hemopurifier from a theoretical concept to being the first medical device to receive emergency use authorization to treat a viral pathogen. In this case, it was a pandemic virus, Ebola. It was also the first therapy to receive 2 FDA breakthrough designations, and it was named a top 25 invention and a top 11 medical breakthrough by Time Magazine. Our Sigyn Therapeutics team is comprised of former Athlon colleagues, and we have since leveraged our industry experience and the emergence of new scientific knowledge and new technology components to create Sigyn therapy. And this is an example of Sigyn therapy, actually an image of Sigyn therapy in action during first-in-mammal studies at the University of Michigan. Beyond accessing a broader range of therapeutic targets, Sigyn therapy has an increased blood flow efficiency, which can contribute to improved treatment outcomes. In our case, the bloodstream, depending on the size of the patient, can flow through the device approximately 16 times during a 4-hour treatment. The device also has an improved capacity to incorporate active components. Again, in this case, we're incorporating a formulation of adsorbent components that have approximately 200,000 square meters of surface area on which to capture our targets. To give that some perspective, 200,000 meters is the equivalent of approximately 50 acres. So essentially 50 acres of surface area in one device to capture targets. Our capabilities expand beyond the capture of viral pathogens. Additionally, our device can remove endotoxin, as well as other bacterial toxins, as well as a broad spectrum of pro-inflammatory cytokines. As a result, we have expansive opportunities in the marketplace. Beyond drug-resistant viruses are the opportunities with drug-resistant bacteria, which is an emerging health threat globally. Endotoxemia, which is one of the leading triggers for sepsis, and sepsis, which is a leading cause of hospital deaths. These capabilities have also established an early clinical and commercialization opportunity to treat end-stage renal disease patients. These are dialysis-dependent individuals. There's approximately 550,000 ESRD patients in the United States. They have an annual mortality rate of approximately 20%, and endotoxemia and concurrent inflammation contribute to shortening the lives of these patients. And once patients have been on dialysis for approximately a year or more, these patients, ESRD patients on average, the incidence of endotoxemia and inflammation is significantly high, in many cases greater than 50%. And so extending the ESRD patient lives, we know would be of value to the dialysis industry. And in this regard, the dialysis industry is dominated by two organizations in North America, Fresenius Medical Care and DaVita. Fresenius has a network of patients of a little over 200,000 patients in North America. And what we look at when we talk about extending the life of patients is what is the value of extending life of patients? And for Fresenius, for each month of extended life for their patient population, that's worth $1.2 billion in additional revenues. And extending the life 1 month, that's only a 1.7% increase in extended life. So if you thought about extending the life by 20% of what would be expected for an ESRD patient, that would double annual revenues for Fresenius. Additionally, these patients are hospitalized quite a bit each year. And so when they're hospitalized, they go out of network. And when they're out of network, Fresenius and other dialysis providers lose revenues. So if you could reduce hospitalization, that allows them to recoup revenues. So in the case of Fresenius, for each week of reduced hospitalization, that equals about $300 million in recouped revenues. Now, if we look at DaVita, extending the life of their patient population by 1 month, again, that's just a 1.7% extension of expected life. That's equal to $960 million of increased revenues and $240 million of recoup revenues for each week of reduced hospitalization. So our ESRD clinical plan, we have an investigational device exemption drafted for FDA submission. The protocol calls for the enrollment of 12 to 15 ESRD subjects with endotoxemia and concurrent inflammation at 3 clinical site locations: San Antonio, Texas, Las Vegas, and Hartford, Connecticut. And our device will be administered during regularly scheduled dialysis sessions. And I will go back to what I said earlier about our device having an increased efficiency of blood processing. This probably wasn't appreciated when I said that, but consider that the optimal blood flow rates of our device correspond with that of dialysis. So that means we can incorporate our device in series with normally scheduled dialysis treatments. So these patients are in their dialysis clinic 3 times a week for 4 hours of treatment. And in our clinical protocol, we're going to include our cartridge in series. And additionally, the protocol design is such where we allow the patients to be their own control. So for the first week, we're going to have the patients enrolled and just look at changes in blood chemistry resulting from normal dialysis treatments. And then in week 2, which is treatments 4 through 6, we'll be quantifying the changes in blood chemistry related to the use of our device in series with the dialysis cartridge. And again, nothing changes in the schedule of these dialysis patients. They have high incidences of endotoxemia and concurrent inflammation, so we think this is a very enrollable study. We look at this as being a modular feasibility study, which you might want to consider from a long-term perspective. When we say modular, this is a feasibility study to demonstrate safety. That's our primary endpoint for this study. But it serves as a demonstration of safety in a very health compromised population, and a study that we can bridge off into, not just to a pivotal indication for ESRD patients, but to other indications in non-dialysis patients. Our secondary objective in this study is to quantify the changes of our therapeutic targets, the changing levels, and hopefully what we will see are significant reductions in endotoxin, as well as a modulation downward of pro-inflammatory cytokine levels and other markers of inflammation. Now I want to talk about oncology. First thing you need to know is we don't develop drugs to treat cancer. We are developing devices to improve the performance of drugs to treat cancer. And I don't think most people think about the tremendous synergy that can occur between devices and drug therapies to treat cancer. Our initial focus is related to chemotherapy. This is the most common class of drug to treat cancer. You might be shocked to learn this, but less than 3.5% of a dose actually gets delivered to tumor cell targets. The remaining off-target chemotherapy contributes to toxicity and long-term health consequences. And tumor-derived exosomes, these are particles released by cancer. They interfere with delivery and they promote treatment resistance. And something you should understand is these particles are in immense concentrations in the bloodstream of cancer patients. In this review article, authored by Raghu Kalari at MD Anderson Cancer Center, he points out that the blood of cancer patients is estimated to contain 4,000 trillion exosomes. Well, 4,000 trillion is the equivalent of 4 quadrillion, which is a 4 with 15 zeros after it. Again, immense concentrations. And so why would you want to remove these particles prior to the administration of chemotherapy? First off, they sequester chemotherapy delivery, and they actually can export chemotherapy that has been delivered to cancer cells back out of those cancer cells. And the circulating presence of these tumor-derived exosomes correlates with treatment resistance. So lower levels of circulating tumor-derived exosomes mean an increased likelihood of patient response to therapy. Higher levels are directly associated with treatment resistance. So our solution is chemo prep. We're looking to reduce the circulating presence of tumor-derived exosomes prior to chemotherapy administration. And our clinical objective is to maintain chemotherapy effectiveness with lower doses, or potentially even increase effectiveness with lower doses. And this decreases toxicity, and it corresponds with the Project Optimus initiative that FDA has recently launched. But consider that if you could reduce the dosing by 50% and maintain the efficacy, that right off the bat cuts toxicity of the patient in half. And ChemoPure, this is delivered post-chemotherapy to clear off-target chemotherapy to further mitigate treatment toxicity. Immunotherapeutic antibodies. As mentioned, these are the best-selling drugs to treat cancer. The market's projected to grow from $104.6 billion this year to almost $464 billion in 2033. This is a field that people feel is revolutionizing the treatment of cancer. This includes immunotherapeutic or immune checkpoint inhibitors, antibody drug conjugates, and other forms of monoclonal antibodies. Well, this past year, Keytruda alone generated revenues of almost $25 billion. One problem is these therapies have severe limitations. Number one, most people don't respond to the therapy. Less than 2% of the doses reach their intended tumor cell targets. This is really bad economics when you consider that a $10,000 dose might only provide $200 worth of patient benefit. And so an issue that we see is that drug decoys that display the binding site of the antibody intercept their delivery. And oftentimes you'll hear people say that antibodies have an exquisite capability to be selective or have exquisite selectivity to bind a cancer target. That's not an accurate statement. They have exquisite specificity to bind a protein or an antigen binding site that's on the cancer target. And unfortunately, that protein can be in high concentrations in circulation and act as a decoy for the antibody. That protein can also be on the surface of exosomes that we discussed earlier. And in this example, this is a publication where the researchers demonstrated that the anti-CD20 antibody rituximab, when administered to patients, when quantified 3 hours after infusion, 50%, almost 50% of the drug was bound by exosomes expressing CD20. Again, consider less than 2% of the drug gets to their target, and again, reinforcing the high concentration of exosomes in the circulatory system. At 3 hours, 50% of the drug was bound. It would be interesting to know how much was bound 6 hours after treatment, but it's an extremely good example of why there's drug decoys that block the delivery of these important therapeutic antibodies. So our solution is Immune Prep. This is a device platform that incorporates an antibody to sweep drug decoys out of the circulatory system prior to the subsequent infusion of the same antibody into the cancer patient. We call this a reverse decoy mechanism. So we want to reduce the circulating presence. We believe, as do others, that if you can reduce the circulating presence of these decoys, then you'll get a greater saturation of the antibody at their intended target. Simultaneously, we expect to be clearing the antibody's therapeutic target to further enhance patient benefit without drug toxicity. So consider that these drugs, their dosing is associated with extensive clinical trials to show what level of dosing is safe. Consider that you could increase the exposure of the body or the exposure of the patient to the antibody without adding drug toxicity. This simultaneous feature does that. So in conclusion, we invent and develop blood purification therapies. Our therapeutic candidates include the Immune Prep platform to enhance the performance of immunotherapeutic antibodies, Chemo Prep to optimize the targeted delivery of chemotherapy. And because, again, as we've discussed, chemotherapy is poorly delivered, ChemoPure is designed to extract off-target chemotherapy from the circulatory system to mitigate treatment toxicity. And our lead product, Sigyn therapy, addresses infectious disease disorders that are not treatable with drugs, some of the most significant unmet needs in global health, and more importantly, a great early opportunity from a clinical standpoint and a commercialization standpoint to treat ESRD patients, which could have a significant impact on the dialysis industry. Some market data on our company. We have just a little over 1.2 million shares, actually 1,263,653 shares outstanding. Recent price $5.25. That puts a market cap value on the company of about 6.6 million. With that, I'll close up my presentation and let me pull up the Q&A box to see what questions we might have. All right, first question. Sorry, this is a bit small. How do you know the dialysis industry will have an interest in your product? Well, what we don't know is to what extent our product will extend the lives of ESRD patients. What we do know is that the dialysis industry does have an interest because we've already worked or collaborated with the clinical research division of a leading dialysis company to establish our protocol. And it was with this group that we were provided our clinical site locations for our feasibility study, and also identified our principal investigators. So we have a lot of work to do still clinically, but the interest of the dialysis industry is certainly there. And I will point out that this is the type of product that the dialysis industry is not likely to develop themselves. DaVita doesn't really have an R&D arm, and Fresenius has been scaling back all new R&D. So, we have a significant opportunity to develop and clinically test and demonstrate effectiveness of a product that could have a big impact on their business. Next question is incorporating an antibody in a device a novel procedure? The answer is absolutely not. We have industry colleagues that have previously immobilized an antibody in their device. They have typically focused on establishing or creating their own antibody to develop the antibody, put it in a device as a standalone therapy. That's a very expensive and long-term proposition that we have seen not work. In terms of the uniqueness or the unique nature of what we're doing, we think it's extremely novel to incorporate an antibody, and we've been filing IP to protect this. But we think it's extremely novel to immobilize an antibody in an extracorporeal circuit to remove circulating drug decoys prior to the administration of the same antibody. And we think this has tremendous synergy. And as I mentioned, simultaneously, we can clear the intended targets without adding drug toxicity. So our approach, we think, is novel. It's highly synergistic. And we think that the establishment of devices that synergistically improve the benefit of drug therapies will be valuable to the drug industry and could be the basis for a new product segment. Bear with me. How competitive is the blood purification space, and can you discuss your IP portfolio? The space is, if you look at the segments of the blood purification space, you have the dialysis industry, which is long established and the best known form of blood purification. Outside of that, things are very segmented within the industry. And what we're doing is interactive devices. Most historically, blood purification has been based on removing things from the bloodstream by size exclusion, filters, but without an active mechanism of action. We actually try to be a little more selective, but need to be broad spectrum in some applications. But we combine with our lead technology the use of filters that provide for size exclusion and then an interactive step to remove and maintain the particles of the therapeutic targets out of the bloodstream, and to do it on a rapid real-time basis. So that industry is quite segmented from an interactive standpoint. Our colleagues at Cytosorbents, they have developed the Cytosorb device, which is well known for its ability to remove pro-inflammatory cytokines from circulation. That is a primary basis of their industry as a public company. Our product does that, as well as perform other features. The Torimixin product, which is endotoxin removal, specifically endotoxin removal, That is a technology that has been commercialized overseas for many years. I believe they disclosed that there's approximately 374,000 treatment experiences that have been completed in humans. The device is actually licensed in North America to Spectral Medical, another public company. And Spectral is advancing the torimixin product through clinical studies right now. It looks like quite a promising study to treat endotoxemia in sepsis patients. And that's a clinical stage product in North America, and that's driving a good market value of approximately 100 million or more. So outside of dialysis, the industry is very segmented, but there's a lot of potential for the industry to evolve to become, a new justified component to treat people. This next question is, how similar are the hardware platforms you need across these four therapies? It's a very good question. And the answer is our lead technology, Sigyn therapy, it has high blood flow rates. And the platform that we utilize are dialysis and CRRT machines that are already located in hospitals and clinics worldwide. Our optimal blood flow rates there are approximately 350 mils a minute is the optimal blood flow. Our other products, as they are focused on treating cancer, these types of blood flows we don't think are feasible to treat cancer. We think you need to have a system in place that can be placed at the infusion center where the patient's going to receive immunotherapeutic antibody therapy or chemotherapy. And we think that's a critical aspect for long-term uptake of our product into the marketplace. So in this case, there are blood processing systems, and these are extremely easy to use. Their blood access is similar to the blood access that's required for blood donation. And again, these are much lower blood flow rates, but very high saturation across active components. And the emergence of these blood processing systems that allow for attachment of an extracorporeal device has really been a critical component to opening up new markets for extracorporeal blood purification technologies, at least the potential to open up new markets just because of the ease of administration. Any joint venture or partnerships internationally? No, not at this time. And, we are really just starting to talk about our opportunities in oncology, and some of the IP that we've been filing, is just starting to publish. So these pending patents, we wanted to make sure we were well protected in place before we initiated discussions. But, we have internationally, we have long-term relationships with the leaders in the dialysis industry. But, we see tremendous opportunities to develop commercialization or partnership opportunities in the oncology field. No, I've read that. So with that, that's our Q&A session. And I appreciate everybody's participation today. And again, I left my contact information on the slide. Historically, from my previous company, we always recognized it was important to have a good interactive relationship with shareholders. And, over time, we would note when we looked at our shareholder list, how many of those shareholders, we knew and develop relationships with over time. So we appreciate your time. Please contact me if you have any additional questions, and thank you very much.